Hello my dear students, myself Meenu Srivastav, and today we are going to discuss about the chemical sterilization. In the gaseous sterilization we have discussed about some chemicals but in the gaseous form, and today we are going to discuss about the chemical sterilization but they are using in the liquid form. So don't waste time, and without wasting time we are going to start our first chemical sterilization, which is the alcohol. Okay, so in the alcohol, ethanol and the isopropanol, or we can say isopropyl alcohol, we can preferably use. Okay, ethanol and the isopropanol, okay, they kill the bacteria and the fungi, but they are not at all effective on the endospores, okay, and non-enveloped virus and they are not effective on the non-enveloped virus also. Okay, they what is the mechanism of action of the alcohol is that they denature the protein and they disrupt the membrane and they dissolve the lipid content of the envelope. Okay, so here the alcohol is in the alcohol, the ethanol is we are using, that is the 70% concentration of the ethanol is the best use concentration. But as in the experimental way, if we are using the 60%, means below the 70%, and the 95% concentration means above the 70% concentration, they kill as a microbial system or microbial flora as the 70% concentration is done. Okay, is doing so. Okay, means the 70% means the 60% and the 95% concentration is doing same as the 70% concentration is doing so. Okay, so most preferable concentration is the 70% concentration. Here the pure ethanol, if we are using for to increase the effectiveness, it seems that the effectiveness is decreased, because the pure ethanol is less effective because it do not help to denature the protein, because for the denaturing the protein the water is essential. Okay, so here the pure ethanol is less effective. For comparatively the pure ethanol, aqueous solution of alcohol or ethanol is used. Okay, now the isopropanol is used mainly preferable than the ethyl alcohol, because it is better fat solvent, or we can say it's a better lipid solvent. So it degrade the fat content or the lipid enveloped present in the microbial flora. Okay, so here it has a more bactericidal action and the less volatile action. Okay, but here except the ethyl alcohol and the isopropanol or isopropyl alcohol, methanol also used, but it's it is used effective against as a fungal spore, but it's a toxic and it's a inflammable. If you have heard about the Jahrili Sharab or the toxic alcohol, that toxic alcohol or the poisonous alcohol is that in the ethanol, methanol is the mixed. In the ethanol, methanol is mixed, so that due to the presence of the methanol, that edible ethanol become poisonous. Okay, so it's a main disadvantages we can say. So advantage, the overall advantage of the alcohol is that it's the inexpensive, non-toxic, and the widely used, widely available, and the rapidly effective. But the disadvantage is that it's not effective against the bacterial spore. It's a main, and in case of the methyl alcohol, or we can say methanol, it's a toxic also. Here the aldehyde, the second chemical agent which we are going to study about, that is the aldehyde. Aldehyde is that two main or mainly preferable aldehydes are formaldehyde and the glutaraldehyde are used. Okay, because and the mechanism of action is that they inactivate the protein by forming cross-linking with the several functional groups present in the protein. Means if in the easy language we want to say that aldehyde, mechanism of action of aldehyde is that they will cross-link the protein groups. The protein groups are like sulfahydryl group, the carboxylic group, the OH group, and the amino groups are present, and these groups are interacted or cross-linked, so the protein nature get disrupted. Okay, now the formaldehyde is commonly available as the formalin. Okay, the formalin is we have studied in the gaseous sterilization, but the formalin use is the preserve of the specimen. Okay, in the biological specimen are preserved in the to preserve the biological specimen, we use the formaldehyde or the formalin solution. And in the bacterial and the viral vaccine, we used in the bacterial and viral vaccine, if we want to prepare the attenuated vaccine, means if we want to inactivate the bacteria and the virus, we use the formaldehyde to inactivate the microbial system. Okay, second is the glutaraldehyde. The glutaraldehyde is the more effective because is the less irritating and the more effective than the formaldehyde. Formaldehyde formula is the SCHO. The 2% solution of the formaldehyde is known as the SIDEX. Okay, but the SIDEX action for the bactericidal and the tuberculocidal is that the exposure only 10 minutes they require. But if you want to reduce the amount of the spores, or if you want to kill the spores, there are 3 to 10 hours exposure is needed. Okay, the advantages, main advantages is that they have the broad spectrum activities, means they kill many of the bacteria, many of the fungus, and but the main thing of the disadvantage is that they are the highly toxic, irritant, and have the carcinogenic property as well. Now come to the next chemical agent, that is the phenol and the phenolics. The phenol structure is that the structure of the phenol is that here. Okay, so here the phenol, Joseph Lister is the first scientist who used the phenol as to sterilize the operation theatre or used as a sterilizing agent. And the phenol synonym is the carbolic acid also, not the carbonic acid. Carbonic acid is the different, and the phenol is named as the carbolic acid also. Okay, but the phenolics, what it means the phenolics? There are some derivatives of the phenol. These combinedly named as the phenolics. Okay, so there are so many phenolics we are using as a sterilizing agent. Here the orthophenyl phenol, the hexachlorophene, and the triclosan. Okay, so the phenols works in the different ways, means they destroy the plasma membrane. Okay, and they denature the protein. Okay, and they inactivate the proteins and the enzymes also. So they act on the enzyme, they act on the membrane, and they act on the protein. So in the different ways they work to kill the microbial system. Okay, so main advantages is the phenol and the phenolics is that these are stable, broad spectrum activities, and the activity of the sterilizing effectiveness, means the effectiveness or the persistence of the effectiveness is that it's for a longer time. Okay, the disadvantage is that they are not effective on the spore. The main disadvantage is that they are not effective on the spore, and second is that it's a disagreeable odor, means the odor is the quite not soothing odor. Okay, and not soothing smell. Okay, and they have the caustic effect. Caustic means the burning effect on the skin. If the phenol 1 or 2 drops fall on your skin, so the skin proteins will degenerate, or we can say the it degenerate the skin protein as well. Okay, so now these have some chemical agents. Now we are going to start the another chemical agents which we have to study, that is the some surface active agents, halogens, and different chemical agents are also there. Now we will study also. Now we will discuss the another chemicals for the chemical sterilization. In this series we will discuss about the halogens, some surface active agents, and other chemicals also. Okay, so now the halogens, as we know in the halogen series, there are the four elements, that the fluorine, chlorine, bromine, and iodine. But in the halogen series, only the chlorine and the iodine is used as the antiseptic or the sterilizing agent specially. Okay, so here mainly the iodine and the chlorine is used as the sterilizing agent. As we know that the iodine is the oldest, most effective antiseptic against all the microbes, fungi, virus, and the bacteria as well. Okay, so how it works with, because it impairs the protein synthesis, means it alter or change the protein synthesis formation, and it alter the cell membrane formation, as well as it combined with the unsaturated fatty acids and the amino acids of the membrane, so it interfere in the membrane formation. So automatically it will degrade the microorganism, because the membrane if the membrane formation will not proceed smoothly, then the microbial system is not at all generated. Now the iodine is present in the form of iodophore. What is the iodophore? Is the iodine plus the organic molecule. Here the iodine is available, it's the oldest material also which is present in the iodine formation. It's a tincture. Okay, in the tincture what components are there? Iodine plus alcohol. But the main drawback of the tincture is that it retains the stain. If we are cleaning any wound, it just give some stain also. Okay? So but the iodophore, which has the iodine plus the organic molecule, in this iodophore the iodine release slowly, so it will produce the sustained release action. And the one major advantage is that it does not produce the stain, and it's a less irritating. Okay. Next is the betadine. What is the betadine? The betadine is the povidone iodine. The povidone is a kind of the surface active agent. So if we use the iodophore with the povidone, it's known as the betadine. Okay, so betadine is a surface acting iodophore. Next halogen is the chlorine. As we know, the chlorine is majorly used in the treatment of water. Okay, so different kinds of the agent which are used, different kinds of the chlorine agent which are used as the antiseptic and the sterilizing agent. The chlorine agents are the hypochlorous acid HOCl and the calcium hypochloride CaOCl2, chlorine dioxide ClO2. The chlorine dioxide is a gaseous form of the chlorine. Okay, it's a gaseous form of the chlorine and used for the area disinfection. Something, if we want to clean the particular area, so the chlorine dioxide, which is the gaseous form, is the most we can say important chemicals. Okay, so it helps to kill the endospore of the anthrax bacterium. Now the chloramine, the other chlorine agent is the chloramine. The chloramine is consists with the chlorine plus the ammonia. The NH3 is the ammonia. It's used as a disinfectant, antiseptic and the sanitizing agents also. The chlorine is mainly used for the sterilizing the particular area for the disinfection, and the water treatment preferably it is used. Okay, now the another chemical for sterilizing agent, that is the heavy metals. What is the heavy metals? The mainly generally silver, copper, zinc and mercury are used as the sterilizing agent. As we know in our rituals, in our tradition, the newborn baby was the first start to feed. First annuprashan is doing with the silver spoon. Means the silver utensil and silver ball and the silver spoon means the first food for the baby provide in that silver ball and the silver spoon, because the silver has a quite antimicrobial activity. That is why they are doing so. Okay, so now you know, now you came to know that the why silver is used, silver katori and the silver spoon is used in the first annuprashan tradition. Okay, the silver, copper, generally silver, copper, mercury and the zinc is used preferably. What they are doing so means how they are doing the antimicrobial, they perform the antimicrobial activity. They inactivate the cellular proteins by binding them. How they binding? They binding with the sulfur hydryl group of the cellular protein. So they produce the interference in the sulfur hydryl group, okay, formation. So the protein will degenerate. Okay, now the silver, that is the silver, it is used as an antiseptic, and the 1% silver nitrate solution used as an antiseptic solution. Okay, and other forms are silver lactate, silver picarate. Okay, AgNO3's name is the silver nitrate. Okay, now it's a bacteriostatic as well as cidal. Cidal means kill, the formation, the killing phenomena. Okay, the copper, now the silver we have completed. Now the comes on the copper. The copper used as the copper sulfate, and it is used, it is a prominent algicide, means it kills the algae. Okay, the effective concentration of the copper sulfate is around 1 ppm. What is the full form of ppm? It's a parts per million. Okay, and the copper 8 hydroxyquinoline, that is a quite antifungal agent. It protect our walls from the mildew formation. The mildew, it's a kind of the fungal infection. So the copper 8 hydroxyquinoline is used in the paint. So it prevent the mildew formation. Okay, now come to the zinc. Different salts of the zincs like the zinc chloride, zinc oxides are used in the different sterilizing agents. Okay, so the zinc chloride, zinc is effective against the anaerobic bacteria, preferably. The zinc chloride is used in the mouthwash formation, and the zinc oxide is an antifungal agent which is used in the paint. Okay, now come to the last chemical sterilizing agent, that is the surface active agent. Mainly the surface active agents, also termed as the surfactant. Okay, the surfactant, how they work, they reduce the surface tension. By reducing the surface tension, they just break the film of either the dust, either the oil layer. So by breaking that layer, they will enter and they will just sweep out of that particular layer. Either it's a oil layer, it's a dust layer, or the germ layer as well. Okay, so here the soap and detergents are act as a surfactant, and they break the oily film layer and they convert into the tiny droplet. It is known as the emulsification process. Okay, so the soap are good degerming agent. Okay, it's not kill the germ, it will remove the germ from the particular surface. Okay, so it is known as the degerming surface. Okay, now come to the different types of the surface active agents. These are acid anionic surface active agents and the quaternary ammonium compounds. As the name states, acid anionic surfactant means the compound contain the anionic group. Okay, the negatively charged group, that is the anionic group. Okay, it reacts with the plasma membrane, and its example is the sodium stearate. The quaternary ammonium compound, that is, it has the positively charged portion, and that portion is known as the ammonium group as mainly. Okay, so that ammonium group strongly bind with the plasma membrane, because the plasma membrane has the positive charged group and the negative charged group as well. So if the acid anionic we are using, then the anionic group combined with the positive charge part of the membrane. And if we are using the quaternary ammonium compound, so the positive part or cation present in that particular compound, it will combine with the negatively charged compound present in the membrane. Okay, so they act on the plasma membrane preferably. Okay, so they have the strongly bactericidal action against the gram positive bacteria, because the gram positive bacteria, the membrane is a particular membrane has the phenomena to bind with these groups. Okay, but it's less effective against the gram negative bacteria. They has the fungicidal, means they kill the fungus, they kill the amoeba, amoebocidal, and they kill the virus as well. Okay, but the main drawback is with it, that is they do not kill the endospore. The example of the quaternary ammonium compound, that is the zephyrin. The zephyrin chemical component is the benzyl alkonium chloride. Okay, and the cefacol, that the next example is the cefacol, and that is the formation, the compound name is the cetyl pyridinium chloride. Okay, here we have completed the chemical sterilization. I hope you have enjoyed the lecture. Any doubt, you can ask me in the comment section. Otherwise, stay safe, stay healthy. Till then, bye-bye. Thank you.